Welcome, everybody. Welcome back. For those of you who joined us yesterday for the first day of the course, but for those of you who couldn't have joined us yesterday, I am Sean Gibbons, a faculty member at the Institute for Systems Biology, or ISB. ISB is a nonprofit biomedical research institute located in Seattle, and we're an affiliate of the Providence Health System. We focus on some of the most pressing issues in human health, including the microbiome, of course, COVID-19, brain health, aging, cancer, and many chronic and infectious diseases. ISB also focuses on improving K through 12 STEM education, and of course beyond K through 12 as well, as you see today, in particular by addressing inclusion and equity for students of color, women, and other historically underrepresented groups in science. You can learn more about ISB by visiting our website, isbscience.org. As a nonprofit organization, we rely on the generous support of donors. We made this event totally free to folks, and we hope that you take advantage of that. But if there's some of you with a little extra cash in your pocket and you want to support a good cause for maybe next year's course, for example, there is a donate button at the bottom of our course website. Before we get into this too far, I'd like to acknowledge that we are on the traditional and unceded lands of the coastal Salish people, past and present, including the Duwamish people. We honor with gratitude the land itself and the Duwamish tribe. I'd also like to acknowledge our generous sponsors who help make these next 3 days possible: Illumina, Metabolon, and the Washington Research Foundation. You can learn more about these sponsors on our website. Because of their support, we've been able to provide this virtual course and symposium free of charge to over 1,000 registrants from over 70 countries. To all of you tuning in around the world, we're grateful for your time today, and we're excited for this opportunity to help train the next generation of microbiome researchers from all around the world. If you're on social media, please remember to share your experiences throughout the 2022 ISB Virtual Microbiome Series using the hashtag #ISBMicro22. And if you want to tag ISB so we retweet you, their handle is @ISBSCI. Okay, so at this point, we're going to launch a poll to get to know a little bit about all of you on the Zoom right now, and I'll keep talking, and then we will sort of review the results of that poll by the time I'm done. So just to give you a quick overview of what we did yesterday, in case you weren't able to join us, our instructor, Dr. Christian Diener, taught us all about amplicon sequencing data analysis using this ecosystem of software tools called QIIME2. And in particular, we focused on looking at samples coming from people from underrepresented groups in microbiome science. Most research in our field, and indeed in most biomedical research, focuses on populations in sort of wealthier urban settings, like places in North America or in Europe. And so we're underrepresented in all this diversity of human phenotypic variation and microbiome variation that exists across the world. It's driven by diet and by culture and all kinds of things. And it's really critical that we understand the span of all of that variability if we want to translate microbiome research into healthcare that is equitably applicable to everyone on the planet, not just to people in North America and Europe. So we decided to focus this year's course and series on populations outside of the US and Europe, and studying them and trying to understand better how to incorporate those data into our work. So we processed 16S amplicon data yesterday from 3 different indigenous populations from around the planet. And today, our instructor, Nick Bowman, who's a graduate student in the molecular engineering program at the University of Washington, is going to walk us through taking these same exact 16S samples and using them to model the metabolic outputs of the microbiota. So we're going to leverage a modeling platform called MyCom that we've developed here at ISB that allows you to build community scale metabolic models of microbiomes where you can modify the community, the composition of what microbes are in the system, and you can modify the diet, the dietary input, and see how that all affects the metabolic outputs of the system. And so today, there's going to be a focus on how diet affects the outputs of the system. So if we change the diet for a given microbiome, how does that affect the metabolites you get out at the other side? And so Nick is going to walk us through that analysis today, which is very exciting. After Nick's course, we will have a talk today by Alex Carr, who's another graduate student in the molecular engineering program at the University of Washington. And he's going to talk about some of his research, not looking at dietary composition of the microbiome, but modulating the composition itself. So if you add an organism that wasn't already residing in a microbiome, can you predict its engraftment if you try to add it into the system? And this is relevant to like probiotics or even infectious disease. If you are exposed to a microbe, will it stick in the context of the commensal diversity of your existing microbiome? So that's going to be a really cool talk that we'll get towards the end of the day. Okay, so that's enough about intro to the course. I'd like to also call out all the people who made this possible. So, in addition to our instructors who've been very visible the last these 2 days, Christian Diener and Nick Bowman. We've also had speakers that are going to be speaking in these 2 days of the course. Yesterday, you heard from Ashish Jha, and today you're hearing from Alex Carr. We have teaching assistants who are on the Slack channel helping you with all your questions. And so those include Noah Rappaport, Alex Carr, James Johnson, Samantha Picos, and myself. Beyond that, we have a lot of administrative support that have helped keep this event going. So our events coordinator, Dominic Lewis, has coordinated across all of our admin to make this all happen with the help of Allison Kudla, Joe Meixter, Audrey Hubbard, Connor Kelly, Victoria Uhl, Shanna Braga, and Thea Swanson. And finally, trying to teach folks about underrepresented populations as ourselves, people coming from overrepresented populations in science, we wanted to be careful not to misspeak or misuse data or not provide the proper context. And so we reached out to Emily Bissell and Dr. Sue Ishak from the Microbes and Social Equity Working Group to be our sort of DEI consultants on this series this year. So they've helped us review our materials and try to build something that is equitable and representative in a respectful way. So beyond that, some people ask about certificates. We will send out an email later on after this series is over to give you details about how to get certificates of completion from having attended this course. If you have any issues with course materials and you need assistance during the course, please visit our course-2022 Slack channel. That's where the TAs will be looking for questions and answering your questions. You've already received a link in the email, but we'll share it again in the chat. So if you can look in the Zoom chat, you should see a link to the Slack channel as well. To ensure that everyone can enjoy the course in a safe and harassment-free environment, we provided a code of conduct, which can be found pinned in the general Slack channel. If we observe behaviors that violate this code, we will warn the perpetrators. We reserve the right to remove problem users from Slack if this behavior persists, but I do not anticipate that this will happen. I just wanted to throw that out there to let you know we have a system in place in case there are any bad actors. All right, so without further ado, I will pass it off now to Nick Bowman, who is going to run the course for this whole day. Oh, and I forgot the poll, actually. Dominic, did it go away? Okay, great, sorry. Now I will read the results of our poll. Okay, so 1% of you are high school students, which very brave high school students doing such a complicated tutorial. Kudos to you for doing this. 3% are undergrads, 46% are graduate students, and 24% are postdocs. So the vast majority of our participants look like grad students and postdocs. 10% of our participants are PIs, like me. So good on you for learning something new, stretching yourself today. Industry scientists make up 5% of our population. And then another 15% are just straight up microbiome enthusiasts, not necessarily scientists, but just microbiome curious and wanting to learn more. So welcome to all of you today. Hopefully you'll get a lot out of the course today. So okay, without further ado, I will pass it off to Nick. I believe we have 15 minutes before the official start time of the course, which is at 9:30 Pacific. So there'll be a little bit of period between when I'm done speaking and when Nick starts off. But you're in great hands today. Nick is a very talented graduate student who has been manipulating these metabolic models and studying the impact of diet on the outputs of microbiomes for a couple of years now. And so he's put together a really beautiful course. So looking forward to an exciting day. Please enjoy and see you at the end of it.
All right, welcome everyone to day 2 of the ISB microbiome course. My name is Nick. I'm a 4th year graduate student in the Gibbons lab, and I'm pleased to be your instructor for today's portion of the course. I'll go ahead and share my slides. Great, so the focus of today's portion of the course is on personalized predictions of microbiome-mediated responses using metabolic modeling. So before we jump into the content today, let's make sure you're all set up with the slides. You can open these slides that we're looking at here using the URL shown. The link should also be in the email that you received prior to the course. And I think the TAs have already posted this link in the Slack channel, so you should be able to find it there. If you have any trouble, just ask in the Slack channel and the TAs will be happy to help you. Quick reminder before we jump in, as with yesterday, this course will be taught across 3 separate modules. The presentation slides that we're currently looking at will contain explanations of the logic and the methods that we're using in the course, as well as links to pertinent materials related to the course today. All the analysis and modeling that we'll be doing, as well as building visualizations of our results, will take place in the course notebook, which is hosted in Google Colab. You can navigate to the notebook using the link on the next slide, but I believe that link is also available in the email that was sent to you, and I believe the TAs have posted that in the Slack channel as well. So you should be able to find it through one of those methods. And finally, as with yesterday, the Slack chat is available to you at any time during the course. If you have any questions or comments, if you have a question, it's likely that someone else probably has the same question. So please don't hesitate to post there, and the TAs will be happy to help you out just as soon as they can. Cool. So before we jump into the slides and talking about some of the methods we'll be doing, let's follow this link here to the notebook and familiarize ourselves there and orient ourselves with the notebook a little bit. So if we follow that link, we'll see this links us to our Google Colab notebook. This looks very similar to the one that you were using yesterday with Christian, but again, the focus of this part of the course will be on modeling microbiota-wide metabolism using Microbiome. So let's orient ourselves a little bit. On the left-hand side, we have our table of contents, which will show you through all the steps that we're using today in order to build and model our communities. We also have our file explorer right here, which will show all the materials that we're using in the course shortly. So before we jump into actually building our models, we'll need to set up the notebook so that we can use it for some of our analyses here. So to do that, we'll need to install some packages that we'll be using for our modeling methods here. A quick disclaimer before we get into the setup process. Microbiome is the package that we're using here in order to build our metabolic models of our metabolic models of our microbial communities. And Microbiome has to do a lot of pretty large and complex linear and quadratic programming problems. In order to solve those, Microbiome requires the use of a quadratic programming solver. There's a lot of really great commercial solvers that are pretty expensive, although they often offer free academic use licenses. But for today's course, we'll be using an open source solver called OSQP, which is installed alongside Microbiome. OSQP is a great quadratic programming tool that works really well for quadratic programming problems, but it's not quite as accurate for linear problems. If you're interested in using Microbiome for your own research, I highly suggest that you look into obtaining a license for another commercial solver like IBM's Cplex or Gurobi, which do offer free academic licenses, as I mentioned. Great, so now that that's out of the way, we can start with setting up our notebook. The first thing we'll do is we will clone the GitHub repository, and this will contain all of the materials that we'll be using for today's course. So let's go ahead and run this cell. Excuse me, I missed a step here. Before you jump into running the notebook, you'll probably want to save a copy for yourself in Drive. So if you go to File and save a copy in Drive, this will open a new copy of this notebook. in your Google Drive. And you can name this something, whatever you like. Great. So this is the same notebook, but now it's just your own copy. And we'll be able to run all the same prompts as previously. Great. So the first step, again, is to clone our GitHub repository. And this will copy all the materials that we need for today's course from GitHub into our file explorer. So let's run this cell. And this should go pretty quickly. So this contains all of the materials we'll be using, including the QIIME artifacts that were built in yesterday's part of the course that contain our taxonomic abundance and the taxonomic mapping that we conducted in yesterday's part of the course. Great, so now that that's done, we can move on into installing MyCom. Installing MyCom is pretty straightforward in Python. We can use pip to install MyCom. And again, this will go ahead and install OSQP, our quadratic programming solver, alongside it. You'll notice that this cell has this exclamation point again in front of our command. And that's just telling the notebook that it wants to run this cell as a command line prompt rather than Python. So this should go pretty quickly. This will install MyCom along with several other packages we need and OSQP. Great, so that's done. And now the last step that we need to run in order to set up our notebook is to install a couple other QIIME2 interactions. So like I mentioned, we'll be using the QIIME2 artifacts that we built in yesterday's part of the course in order to build our metabolic models within MyCom. In order for MyCom to be able to interact with those QIIME2 artifacts, we'll need to install these two packages as well. And now this should go pretty quickly. But while that's running, we'll switch back over to our slides and we should be able to see, well, we'll investigate some of the methods and processes that we'll be using today. Great. So yesterday, if you recall, we used QIIME2 to analyze sequencing data, starting from raw data and then using that to examine diversity metrics and map to taxonomy, among some of the other methods that Christian introduced. Today, we'll be looking rather at functional analyses of the gut microbiome. That is to say that rather than looking at what populates the microbiome, we'll be determining what is happening in the microbiome, metabolically speaking, which is not always fully explained by taxonomy alone. So when I first say functional analysis, a few things might jump to mind, like some of the omics methods that are listed here, metagenomics, metatranscriptomics, for instance. Most commonly, to determine some of the function of the microbiome, one might do a metagenomic study, from which you can see what genes are present in the community, map those to reactions, and determine what sort of pathways might be active in the community that's being modeled. There is a caveat, though, that in gene abundance data doesn't always tell us the full story that we're looking for. And so that's because gene abundances really only report to us the metabolic capacity of a community. If you imagine a microbial community with really high abundance of starch degradation genes, you might then infer that a lot of starch degradation pathways are active in that community. But if the host doesn't eat starch, then this is clearly this inference is incorrect. So gene abundances tell us what could happen, not necessarily what is happening. And in the case of functional analyses, we really are interested in what's actually happening biologically in the community that's being modeled. So one school of thought that we'd like to present here is that gene abundances are really cool and we can learn a lot from them, but they don't necessarily tell us the whole story that we're after. So today we're going to explore an alternative approach that one can use in order to analyze the function of the microbiome, which is predicting metabolic flux. And fluxes actually will tell us more about the actual metabolic behavior of the microbiome than abundances are able to. Great. So let's talk a little bit about fluxes, what they are and why we care about them. So rather than abundances, which are a measure of concentration, fluxes are a measure of mass conversion into a system and through internal reactions within that system. Since they represent a rate rather than a concentration, Fluxes are typically measured in units of concentration per time, for instance, millimoles per hour. In bacterial communities, like the ones we're modeling today, we typically also scale flux by relative abundance in order to account for differential metabolic contributions by more or less abundant taxa in the community. All right, so why do I claim that fluxes are more informative than abundances? Well, if we look at the example on the right-hand side, we see a cell, we see glucose going into glycolysis and then into the TCA cycle. And we see that the cell in question here is importing a lot of glucose. But that glucose is almost immediately being converted into pyruvate as it enters the TCA cycle. If we were to measure the internal glucose levels of the cell, we might see that they're actually super low. And if this was the only measurement that we're taking, this might lead us to incorrectly assume that glucose is not important for the growth of the cell, which obviously we know is not the case, because we can see that a lot of glucose is important in order for the cell to grow. This might seem like a pretty naive assumption, but if you imagine all of the different fluxes occurring in a metabolic community, you can see why looking at fluxes might be more informative than looking at abundances. They tell you not only which pathways are active, but also which precursors are important in order to activate pathways later on. There is, of course, a problem with measuring fluxes, as there always is. And that problem is that they can be very costly and laborious to measure. One thing we could do is longitudinal metabolomics, wherein we take metabolic abundance measurements at consecutive time points, and then use those measurements in order to calculate a rate of conversion between the time points. But this takes a lot of effort, is fairly expensive, and is only limited to those metabolites that are included in our metabolomic panel. Another option is isotopic labeling, wherein we feed a community, for instance, glucose with isotopically labeled carbon, and then we measure how much of the resulting pyruvate includes those isotopically labeled carbons. From that, we can then infer a flux that's occurring in the community. But again, this is even more costly than longitudinal metabolomics, and is still limited to just those metabolites that are being targeted in our panel that we're conducting. So really what we need is a way to easily and accurately estimate fluxes without the labor and expense associated with these methods I've just presented here. Luckily for us, there is already a computational tool that does that. So to do just that, we can use a method called flux balance analysis, which has become more and more popular in microbiome research in the previous years. Flux balance analysis is a computational method that uses genomic information, like the information that we got out of CHIME yesterday, to infer the most likely set of fluxes in a biological system. We can use this to predict the modeling behavior of a microbe using genome-scale metabolic modeling. So let's take a look at how flux balance analysis works, some of the assumptions it makes, and how we can use this tool in order to infer fluxes in our system that we're modeling. So like I mentioned, FBA is a powerful computational tool that allows us to infer fluxes through reactions in a system by reducing the possible solution space of our fluxes to one that's biologically relevant. So that means that it's a solution that fits what's actually happening in the system that we're measuring in vitro. FBA makes a couple of assumptions, the most critical being that the system being modeled, in this case, the metabolism of a single microbe, is in a state of constant flow or steady state. This just means that the inflow and the outflow of reactions are balanced. A good analogy to this is imagine opening a window in your house. You'll have air flowing in and air flowing out, but you won't suddenly have more air flowing in than out. Suddenly your house expands and pops like a balloon because the air pressure in your house has suddenly increased. That doesn't happen. So the steady state assumption mimics this, and it's actually been found that this is pretty applicable to a lot of different biological phenomena. If we look at our example reaction over here, we have hydrogen and oxygen combining to form water. In steady state, we can say that stoichiometrically, the amount of hydrogen and oxygen consumed on the left hand of the equation is equal to the amount of water produced on the right hand of the equation. We can represent this mathematically in our balance equations down here. We have our products with a positive coefficient, our reactants, hydrogen and oxygen, with a negative coefficient since they're consumed, and we set the balance of this equation equal to 0. If we repeat this process for all of the reactions in our system and factor out the fluxes, we're left with a matrix called a stoichiometric matrix that represents all of the reactions present in our system that's being modeled. We're also left, the stoichiometric matrix is designated S in this equation here. We're also left with a set of unknown fluxes designated V, for which we're trying to solve. To satisfy the steady state assumption, we can set the dot product of our matrix S and our unknown vector of fluxes V equal to 0, and this sets up our system of linear equations. Additionally, we can add constraints to the individual fluxes within our system. For instance, we can set the lower bound of this flux here, water, equal to 0. This will force the production of water, thereby adding flux through the first equation. Solving for our set of equations that satisfy the steady state assumption and our additional constraints leaves us with what's called a flux cone. The flux cone is the space in multidimensional flux space that includes all sets of fluxes that satisfy our constraints and the steady state assumption. You'll notice that the 0 point here is also included, and this is called the trivial solution, wherein all fluxes are set equal to 0, which satisfies our constraints, but isn't necessarily biologically relevant. You might notice that this flux cone sort of feathers out from 0 and actually includes an infinite set of solutions that satisfy these constraints. Now, obviously, we can't do much with an infinite set of solutions. So what are we doing next? Well, the overall goal of FBA, flux balance analysis, is to reduce that flux space to one that is biologically relevant, that, excuse me, that mimics the actual behavior of the system as well as possible. So how do we find that optimal solution in the solution space where we have an infinite number of solutions? Well, first we can consider growth rates. We also make the assumption that if a given microbe is measured to be present in a system, it must be able to grow. If it weren't able to grow, it would die off, be excreted from the system, and we wouldn't have measured it there in the first place. So if we say that it's able to grow, in practice, this means that we can say that we want a solution in that flux cone that corresponds to maximum biomass production, since this is typically the biological goal of a growing microbe. Biomass production can also be represented as a flux, since it is, in essence, a measure of mass conversion. What's neat about flux balance analysis is that we can add information from a number of different sources in order to determine our optimal solution. As we've mentioned, we can use the reactions that are present in our system to build a stoichiometric matrix, like the one that we described in the last slide, that mathematically represents all the reactions taking place in our system and how they interact with each other. For instance, what precursors from one reaction are required in another. We can actually infer this whole stoichiometric matrix from the genome of a microbe by looking at gene abundances and then mapping those to reactions that we know are taking place in that microbe, from which we can then build a reaction network leading us to our stoichiometric matrix, which we set up as our set of linear equations. Another thing that I've already mentioned is that we assume that the system, the microbe that we're modeling, is at steady state. And so in practice, what this means is that we're modeling the microbe in the exponential growth phase. Because in this growth phase, the growth rate is constant, and therefore biomass production is constant. So we include this assumption to be able to arrive at a solution for our fluxes. We can also include thermodynamics in our solution. For instance, if we know that thermodynamically a reaction can only occur in one direction, we can set the flux of that reaction, the lower bound of the flux of that reaction to be equal to 0, forcing it to only occur in one direction, the forward direction. Finally, we can also add constraints to the system. We can also add constraints to the import of metabolites going into the system, which we can leverage to represent the metabolites available to that system in order to grow. In practice, this basically allows us to model the diet that's available to the microbiome, adding relevance to the predictions that are being made by flux balance analysis. Adding all these constraints in and then solving for our fluxes, we can find that region within the flux cone that corresponds with maximal biomass production, represented by this red band here. So this is great. We now have a subset of our solution space that corresponds with the overall evolutionary goal of this microbe that we're modeling, which is to maximize its growth rate. However, you might notice that there still is an infinite set of solutions within this within the subspace of our flux cone. So the real art of FBA is to then select a single solution in that subset of solution space that's the most biologically relevant. Again, mimics the growth behavior of the microbe most closely. There's several methods available to us to find that single solution within our subspace. But today we'll focus on these last two listed here, since these are ones that are leveraged by Micom in order to find a solution. One such solution is to minimize the imports into a system. Since the microbiome can be a highly competitive environment, this is biologically intuitive, since the most successful residents of the microbiome will not be relying on importing lots and lots of different metabolites in order to survive. And rather, they're able to subsist on a smaller subset. Another common approach is parsimonious flux balance analysis, sometimes called PFBA, which basically minimizes the number of enzymes required to satisfy the constraints of our flux balance analysis. PFBA has actually been found to reproduce experimental fluxes in E. coli very well. And intuitively, this makes a lot of sense, since bacteria are very efficient beings that don't like to produce more enzyme than they need to in order to grow. So really, the method that you should use in order to find your solution in your reduced subspace is related to the question that is being asked and the system being modeled. For instance, is it competitive? Is it resource limiting, etc.? I encourage you to look more into these methods if you're interested in conducting your own flux balance analysis in the future. So to this point, we've been focused on how flux balance analysis can be leveraged in order to simulate genome scale metabolic models. So how we can determine the fluxes in a single bacterium. To make metabolic models of microbial communities or metagenome scale metabolic models, we use a tool called Micom, which extends flux balance analysis into microbial communities. So to first build these models, we need to pass in the relative abundance of the bacteria in our sample. Due to differences in sequencing efficiency, there may be some bias towards some bacteria over others, but the abundance from our sequencing data should be more or less representative of the community that's being modeled. Micom will then map the abundances of these, will then map the abundance data to a database containing hundreds of genome scale metabolic models for common gut microbes. So basically a database that contains hundreds of reconstructions of individual taxa that are commonly found in the gut microbiome. Micom then uses these reconstructions of the taxa that are present in the sample to build a massive stoichiometric matrix like the one that we described earlier, that includes not only the internal reactions within each taxon, but also exchanges between taxa and external reactions, basically imports and exports out of the community to the external environment, which in this case is the gut lumen of the host. After this, we can then specify particular diets that basically indicate what metabolites are available to the microbiome in order to grow. Micom will then use FBA with an additional regularization step that we'll talk about in upcoming slides in order to calculate unique growth rates for most of the taxa present in our community. Using these growth rates, Micom then estimates fluxes that are occurring within this community and returns those as predictions. So all in all, this gives us a huge amount of output data that we can use, that we can then use to make detailed and interesting predictions about imports and exports into and out of our microbial community. It also tells us about the sort of inner network of reactions occurring within each bacteria that it's typically very difficult to measure. It can tell us about codependencies between bacteria. So for instance, if one bacteria is dependent on metabolites produced by another or inhibited by metabolites from another, we can model that in these metagenome scale metabolic models. And it also provides us with sort of a testing ground for potential interventions. So if we want to see how different dietary contexts affect the metabolic output of the microbiome, we can do that with these metagenome scale metabolic models in MyCom. So here's a quick overview of sort of the typical workflow within MyCom. Like I mentioned previously, we start with feeding that abundance data in, as well as a database of genome scale metabolic models. MyCom then maps the abundance data to that database and pulls in reconstructions of all the taxa present in our model. We then apply a medium and allow MyCom to predict unique growth rates for most of the taxa present in our community, and then also predict fluxes based on those growth rates that are then output to the user. We can then use those predictions for growth rate and exchange fluxes in order to build visualizations and interpret our results. What's really neat about MyCom is that it was built to be supported by two separate interfaces. There's the Python interface that we'll be using today in our Google Colab notebook, but you can also run all of these commands directly in QIIME 2. So if you start your pipeline in QIIME 2 and would like to run the whole MyCom pipeline there, you're able to do that. What's also great is that it's very easy to switch back and forth between the two interfaces. So if you start your MyCom build process in QIIME 2, you can then take those QIIME artifacts that are produced and bring those into the Python interface and continue the pipeline there. So this is a really great tool that's very extensible and easy to use. Great. So now that we have a basic idea of flux balance analysis and a quick overview of MyCom, let's switch back over to our notebook and we can take a look at how we'll go about building and growing our communities. Right, so all of the setup is now complete for our notebook here, and we can jump into actually using MyCom. So again, here's just a quick overview of the MyCom pipeline, if you'd like to familiarize yourself a little bit more. And like I mentioned in the slides, the process of building metabolic models in MyCom begins with constructing a combined abundance taxonomy table that after this point, we'll just refer to as a taxonomy table for simplicity. Before we load in our own data, we can import an example taxonomy table directly from MyCom and take a look at what that should look like so we can compare that to our data that we constructed yesterday in QIIME 2. Let's run this cell and take a look at the output. Cool, so this is what a typical taxonomy table for use in MyCom should look like. You'll notice in this table, we have 4 identical strains of E. coli spread across 2 different samples, sample 1 and sample 2. We can see that each row here represents a single taxon within a single sample, and that we have a few different columns here, each of which are important. So there are 4 required columns in a taxonomy table in MyCom. First, we need this ID column, which basically specifies the unique identifier for each taxa in our community models that we're building. We then also need a column specifying the lowest rank to which we're building the lowest taxonomic rank to which we're building our community models. In this case, we see that we have the species rank, indicating that we're building models at the species level. MyCom will use the name of the species here in order to map to that model database that contains all of the individual reconstructions of taxa that are being used to build the metagenome scale metabolic models. If we include higher order taxonomic ranks, like for instance, here we have genus, MyCom will use those in order to map to the database, but they aren't required. So any additional taxonomic information you pass in will be used in order to map to the database. But again, it's optional. And in fact, if your database and your taxonomy table are annotated differently, this can lead to situations where maybe a taxon is named differently in the database than you have in your taxonomy table and is then omitted from your communities as a result. So just be very careful about how you're naming your communities in your taxonomy table and make sure it's consistent with the database that you're using. We also see that we have a sample ID column. This is required to tell MyCom which sample each of these taxa is residing in. And then we have an abundance column, which in this case shows us read counts and just tells us how abundant that taxon is in that given sample. Cool. So you might notice that, yesterday we built tables in Chime. And so that's not necessarily so easy to read in, but no worries, we can deal with that. Like I mentioned previously, MyCom is built in order to be able to accept Chime outputs. And that's why we installed those two packages early on, allowing us to take a look at Chime data directly in MyCom. So we will import our data here shortly, but before we do, I'd like to touch on that model database that I've been alluding to a few times throughout this course. So basically what we need in order to build our MyCom models is a database that has pre-curated reconstructions of individual taxa. And then MyCom will scrape that database for the taxa that are present in each one of our samples and use those individual reconstructions in order to construct our metagenome scale metabolic model. So we'll be using the Agora database, which is a curated database of more than 800 bacterial strains maintained by the Thiele lab. And these are strains that commonly live in the human gut. In particular, we'll be using a version of this database summarized at the genus rank, since we will be building our models at the genus level, since that is the lowest taxonomic rank that is we can get reliable information from 16S data, like Christian mentioned yesterday. This Agora database can be downloaded from the MyCom data repository if you're interested in using it further. But we also copied that over from our GitHub repository earlier, so we don't need to worry about downloading that ourselves. It should be in our materials folder on the left-hand side in your file explorer. You see it right here, Agora 103_genus, and that's also a Chime artifact. Great, so now we have that out of the way, we can get into loading up our own data and building our taxonomy table that we'll use in order to build our communities. So to do this, we'll import this Chime to MyCom function from MyCom, which will allow MyCom to read in our Chime artifacts, specifically our table artifact, which contains abundances, and our taxa artifact, which contains our taxonomic mapping. We'll also include this collapse on argument, which summarizes the abundance data to the genus level. Since again, we're building our models on the genus level. So we'll run this, and this will go pretty quickly. And again, we only had to specify the genus level since that was what we were using to build our, since that's the level that we're building our models for. Great, so we can now take a look at the output from our Chime to MyCom function and take a look at the taxonomy table that was generated. Awesome. So this looks a lot like the example table that we looked at just a second ago. We have our sample ID column, which specifies the individual sample that a taxa is present in. We have an abundance column telling us the read count of that given of that individual taxa in that sample. We have our lowest taxonomic rank summarized in this table here. We see that we're using the genus level, so we have individual genera names here. We also have an ID column, which again is a unique identifier for each one of these genera. However, you'll notice that in this case, the genus column and the ID column are identical, and that's okay. It doesn't have to be, but that works just fine. We also have a relative abundance column here, which basically just gives us abundance again, but this time in compositional form as relative abundance rather than absolute read count. Awesome. So now that we have these, our taxonomy table ready to go, we're theoretically ready to build our models. But before we do that, we'll do one more thing, which is merging our taxonomy table with our metadata. So if you recall yesterday, we had metadata available for each one of our samples. We will merge these tables together, and that way we'll have it on hand for following steps. So we can always go back and reference the metadata in upcoming steps. So if we run this and then take a look at our output, we see that we still have our same taxonomy table, but now we've merged it with our metadata. So in addition to abundance, we also have ethnic group, subsistence type, and country of origin for all of our samples. Cool. So with our taxonomy table ready to go, and now our metadata is merged, now we are really ready ready to start building our models in Micom. Cool. So now that we've really set ourselves up nicely, building the models is fairly straightforward and really should be a breeze. So in order to do this, what we will do is import this build command from the Micom workflows module. And then in this build command, we'll start running this now, and then I'll talk through a little bit of what's happening in the background of this build command and some of the inputs that we put into it. So in this build function, we'll pass in tax, which again is our taxonomy table that we just built from our CHIME artifacts we constructed yesterday. We'll pass in the name of our model database. So again, this is the Agora database containing genome scale reconstructions of several hundred common gut microbes, excuse me. We'll also pass in the name of the model folder that we'd like to have our models saved to. So as Micom goes through and builds individual models, it will save these models in a serialized form in the output folder that we specify here. So you'll see that now on the left-hand side in our file explorer, we now have an output folder called models, and that's where all of our models will live. We'll specify the solver we're using, which again is OSQP. If you were to get an academic license of a different solver, then you could change the solver that you're using, and you might get better numerical efficiency. This cutoff command here specifies the relative abundance threshold that we'll use in order to build our models. So in this case, our cutoff is 0.01, which designates 1% relative abundance. And so what that means is that any taxon present in our sample at less than 1% relative abundance will be omitted from the models. Now we do this in order to, since we assume that taxa present in that low abundance aren't substantially contributing to the metabolic output of the models. I do urge you, if you are planning to use Micom on your own, to take a look at that cutoff and determine what is a good cutoff for your own analyses. Typically, we'll use a slightly less strict cutoff, but for the purpose of this course and to minimize the sort of computational intensity that was required, we elected to go with a 1% relative abundance cutoff, and that seems to work just fine. We'll also parallelize this process over 2 threads, which is what this command does here, and that'll minimize the amount of time that's required in order to run this command. Great, so this all in all should take about 10 minutes to run, and so while that's going, we'll switch back over to our slides, and we'll discuss a little bit more about the next step in the Micom process, which again is is growing the models that we're building here. Great. So now that we've covered like classic flux balance analysis that we use for genome scale metabolic models, so I think the metabolism of one single microbe, everything seems pretty straightforward. You just maximize the growth rate, you find that maximal biomass subspace within your solution space, and you get your answer for your fluxes. So can't we just do the same thing for community models? And the answer is not quite. So in a genome scale metabolic model, for a single bacterium, we only have one growth rate, which we can designate mu. In a microbial community, however, we have several growth rates. So we have mu sub I for each individual growth rate, for each individual taxon in our community. And we also have an overall community growth rate designated mu sub C. So mu sub C can be calculated by summing together the abundance of each individual taxon multiplied by its own individual growth rate. So could we just maximize the community growth rate and then get an answer that way? Well, not quite. If we try to just maximize community growth rate, you'll find that we pretty quickly run into trouble. And we'll discuss why right now. So in practice, getting a community-wide growth rate using just classic flux balance analysis doesn't work very well. Let's set up an example here to illustrate. where we have two identical microbes in our community. We can say they're both E. coli, and they're completely identical, except for their different colors. And let's say we have them in the exact same abundance, so 50% relative abundance for each. If we know that the maximal community growth rate is 1, we can see that there's actually an infinite number of solutions to this. We could have both of them growing at a growth rate of 1, which after we multiply by relative abundance, still comes out to a community growth rate of 1, based on our definition in the last slide. But alternatively, we could also have a growth rate of 2 for this blue E. coli, while the orange E. coli is not growing at all. This would still give us a community growth rate of 1, despite the fact that 50% of the taxa present in our community aren't growing at all. So this plot here actually shows us this. We have this red line representing our optimal maximal community growth rate, and we can see that there's an infinite number of solutions on this line that satisfy that maximal community growth rate, including these terminal solutions here, wherein one of our taxa is growing at a high growth rate, while the other one is not growing at all. So this isn't good, since this introduces a lot of ambiguity to our system, and it increases the size of the flux cone to include non-biologically relevant solutions. And that's obviously not what we want. Intuitively, we know that if we have two identical strains, they should be growing at about the same growth rate. And if we make the assumption that we mentioned earlier, which is biologically intuitive, that any taxon present in a sample should be able to grow, we shouldn't include these solutions wherein one taxon is growing and the other isn't. So to overcome this, MyCom introduces a regularization technique that reduces our solution space back to one that's relevant. So again, if we make that assumption that anything present in the sample should be able to grow, the regularization strategy should return us a solution in which most taxa are growing, omitting those solutions wherein one taxa is growing at a fast rate and the other one is not growing at all. What's called an L2 regularization scheme actually achieves this pretty well. And it does this by minimizing the sum of the squares of the individual growth rates. Basically, what this does is penalize those solutions wherein one growth rate is really high, and it distributes growth back across as many taxa in the community as possible. And indeed, we see that if we apply this L2 regularization scheme to our example community here with our two identical E. coli, it finds an optimal solution wherein both taxa are growing at the exact same growth rate with a growth rate of 1. And in fact, L2 regularization will always find a unique solution along that line of maximal community growth rate that tries to distribute growth as evenly as possible across taxa. You might, though, if we approach this biologically, though, it does make sense that for all the bacteria to be able to grow in a community, optimal growth might not actually be able to be achieved. An idealistic maximal growth rate might only occur when some really fast-growing bacteria are growing at the expense of other slower-growing bacteria. So in MyCom, we can model this by incorporating a cooperative trade-off parameter. And basically what this does is specify the fraction of the maximal community growth rate to which we want to perform our L2 regularization. So for instance here, if we set a trade-off parameter of 0.8, so 80% of maximal community growth rate, we see that our L2 regularization finds that optimal solution at 80% of the maximal community growth rate, lowering our growth rate slightly. And you'll see in the next slide why this is sometimes more biologically relevant than that optimal solution along that maximal community growth rate line. Methods for choosing a trade-off parameter are covered at the end of the course notebook. So if you're interested in performing your own analysis, MyCom, I urge you to take a look at that after the course is complete. So cooperative trade-off flux balance analysis, like we just covered, allows us to treat those metagenome scale metabolic models that we're building with some of the same methods that we do for genome scale metabolic models. So again, we can use parsimonious flux balance analysis, for instance, or minimization of imports in order to find a unique solution within our flux space. But the age-old question, does it actually work? Does cooperative trade-off flux balance analysis actually give us answers that are biologically relevant? So here on the left, we can see the results from a sample MyCom community with the number of taxa growing at various trade-off values. For reference, a growth rate less than 10 to the minus 6 is effectively no growth at all. So we consider this gray shaded region to be taxa that are not growing in the community. We see that in this community, when it was grown without an L2 regularization scheme, we see a very small number of taxa able to grow at an appreciable rate, with almost all of the taxa present in the sample not growing at all. We then see that if we do apply our L2 regularization scheme, still optimizing to that maximal community growth rate, we see distribution of growth across many more taxa. So we see many more taxa that are able to grow in that region to the right of the gray shaded region. We still see some taxa that are not able to grow, but growth is distributed much more evenly across the taxa. We then see that lowering that trade-off value to about 0.7, so performing our regularization strategy to about 70% of the maximal community growth rate, we see that almost all of the taxa are able to grow, with only a few taxa not able to grow. So this actually already satisfies the assumption that we made previously, which is that taxa that are present in the sample should be able to grow. And this sort of echoes what we're expecting to see in an actual microbiome, which is that most of the taxa are growing. So this in and of itself is already an indication that the L2 regularization scheme with cooperative trade-off seems to be giving us more biologically relevant results. The next thing the team wanted to answer, though, is are those growth rates that are being predicted by MyCom actually relevant to what we see in the communities? So for these data that they were using, the team had metagenomes, and it's been shown by the Segal lab that coverage profiles of metagenomic reads, like the one seen here, can be used in order to estimate a growth rate. You can do this by measuring the decay and read coverage from the origin of replication as it moves away, and from there infer a growth rate in fast-growing taxa. So using this method, the team could actually measure growth rates in vitro using this replication rate estimation method and compare those to the growth rates that were predicted by MyCom. So what we see is that without the L2 regularization scheme, or with the L2 regularization scheme, but still maintaining that maximal community growth rate, there was very little correlation between actual growth rates measured in vitro and those returned by MyCom. Again, the team saw, though, that bumping down that trade-off value, so optimizing to 70% of our maximal community growth rate, actually returned growth rates that were much more correlated with what was seen in vitro. So really what this tells us is that cooperative trade-off flux balance analysis satisfies our assumption that taxa that are present in the community should be able to grow, and it returns to us growth rates that are actually biologically relevant as compared to other methods without L2 regularization. This also shows us that optimizing to that maximal community growth rate, even with L2 regularization, isn't always the correct technique, and that sometimes sort of a suboptimal growth rate is the one that's actually the most biologically relevant. So that trade-off value is actually very important moving forward as we build and grow our models in MyCom. Cool. So all these methods seem great, and all the math seems to check out. So what's taking so long to run all these workflows? Well, typical MyCom models, like the ones that we're building here today, are actually a bit more complex than the examples that I've been using to illustrate the methods, like, for instance, our 2 E. coli community. And actually, we can take a look at that here. So this is a visualization of a standard metagenome-scale metabolic model that you might build in MyCom. So each pink dot in the visualization here represents a single metabolite, and each blue dot represents a reaction. And those reactions and metabolites are connected with black lines. you'll see that each of these regions that seems to be a little bit distinct from the others represents one taxon. And there are thousands of internal reactions as well as exports and exchanges between individual taxa and with the outside environment. So you can see that this community model shown here has nearly 70,000 reactions, over 45,000 metabolites, and nearly 300,000 unique connections between those. So you can imagine that finding an optimal flux solution through this model takes a little bit of computational power and sometimes a little bit of patience. So it can take a little while to run these models, but the output from MyCom gives us an immense amount of data that that finds sort of the most biologically relevant flux through this massive model that's shown here. Awesome. So now that we have a better understanding of some of the methods that MyCom and assumptions that MyCom makes during the modeling process, we can switch back over to the notebook that we've been using. The model should have built at this point, and we can move forward with the next step in the process, which will be growing our models, and from that, developing an output that we can use to build visualizations of our results. So we'll switch over to the notebook. And hopefully at this point, your models have completed building. If not, please go to the Slack and the TAs will be able to help you there. Before we move past this cell that we just ran, I want to call your attention to this warning that was given underneath the cell that we just ran, wherein we built our models. So you'll see this warning that says less than 50% of the abundance could be matched to the model database. Model Hadza3 may not be representative of the sample. So what this means is that MyCom took the taxa present in this model and tried to map them to the model database, the Agora database we're using, and it wasn't able to find reconstructions for more than 50% of the abundance of the taxa in that model. Typically, this would be some cause for concern. Typically, a fraction of 80% or more is considered great, and we really shoot for greater than 50% abundance matched to the model database. However, in this case, this is one of the issues that we face when modeling underrepresented groups in microbiome research. The database that we're using, in this case, the Agora database, has been built based on microbiome studies of overrepresented groups, namely European and North Americans. So updating these databases to be representative not only of those individuals, but also of these underrepresented groups like the ones we're modeling today, is one of the things that the microbiome research community needs to address in upcoming studies in order to make microbiome research more equitable and applicable to individuals all around the world, and not just in those overrepresented groups in North America and Europe. All right, so with that out of the way, let's take a look at the output from our build command. I do want to mention that we will continue with this model, even though less than 50% of the abundance could be matched. But it is an important thing to keep an eye out in future projects. Again, it is something that the microbiome community needs to address in coming years. So the output of the build command gives us this file that we've named manifest. And so what we see here is, again, our sample IDs, as well as the metadata that we've passed along into the build command. And on the right side of this data frame, we see our statistics from the build command. So this found taxa column designates how many of the taxa in the sample were mapped to our database. The total taxa is just the total number of taxa that were present in the taxonomy table that we passed into the build command. The found fraction is very easily just the found taxa divided by total taxa. And then the found abundance fraction is that found fraction weighted by relative abundance of taxa in each one of these samples. So we can see that most of these seem to be above 70% at least, which is pretty good. And then we see a few like this one here that's below 50% that don't have quite as many taxa mapping to the model database. And that's all right, we'll continue with that. But that's a good thing to keep an eye on when you're building your own MyCom models. Great. So now we have our community models built, and now we can leverage MyCom to conduct the next step, which is simulating community growth. But before we jump into that, let's discuss a little bit what it is we want to look at with our MyCom models. So let's think a little bit about the insights that we might want to gain from these samples. So first and foremost, we can use MyCom in order to investigate the metabolomic response of the gut microbiome. And we've discussed that a little bit. And one thing we're interested in is how that metabolic output changes based on dietary input. So we can use MyCom to take a mechanistic look at how the dietary input changes the metabolic behavior of the microbiome. So for instance, if we were to take individuals from an indigenous population, like the ones that we've been modeling here today, and we were to simulate the growth of these models on a matched diet, so a diet that's representative of a typical meal eaten by an individual from that population, and then simulate it again on an unmatched diet. For instance, today we'll be using a representative Austrian diet. How does the metabolomic profile of the microbiome that we're modeling shift? What exports are different? How does the growth behavior of each of the individual taxa change based on the dietary input that we feed the microbiome? So that's sort of the overview of what we plan to do with our models here today. So as I mentioned previously, before we can conduct the growth simulation, we need to pass in a medium that designates which metabolites are available to the microbiome in order to grow. So to do that, first we need to load our medium. So in this cell, we'll download the matched diets for each of the 3 communities that we're studying that was constructed by our team by hand. This is a CSV file, so we can easily read it using pandas. So we'll run this cell. And if we actually take a look at the output from this, just type this, and we look at the output, we'll see that this data frame is pretty long. And that's because the medium shown here is per sample, which means that each of the 9 models that we've built in the previous step has its own medium represented in this file, which is dependent on the food typically eaten in the community that they live in. So we have actually 9 separate media represented in this file, along with sample IDs, so they will be applied only to those taxa, only to those samples that they correspond with. Great. So now that we have our media loaded and we have our models built, it's time to actually grow our models. So again, we have 9 individual samples that we are using, and we have this per sample matched medium. So we've actually set ourselves up very well, and this grow command will be, again, pretty straightforward and easy to use, but it will take some time. So I'll start running this cell here, and I encourage you to do the same. And then we'll discuss a little bit about what's going into this command. So again, from the MyCom workflows module, we'll import our grow command, as well as our save results command. We'll then run this grow command, which will take the models that we've built and it will apply cooperative trade-off flux balance analysis in order to estimate unique fluxes for most of the taxa present in our community. And from there, also estimate fluxes happening in each of these taxa, and report those back out to us. So in grow, we will pass in our manifest that we got out of the build command previously. We'll specify the folder that we saved our models to. So if you recall, we saved all of the models in serialized form in this models folder shown on the left-hand side in your file explorer. We'll designate the medium that we want to use, which again is this matched medium, which is representative of a typical meal in each one of the indigenous communities that we're modeling today. And we'll also specify a trade-off value, which, if you recall, is the fraction of the maximal community growth rate to which we're running our L2 regularization scheme. In this case, we'll use a trade-off of 0.5. This seems to give us pretty good results, but if you are to run your own MyCom models on your own time, I do encourage you to take a look at the end of this notebook at the trade-off analysis section, which will help you understand a little bit better how we go about selecting a trade-off value in MyCom and moving forward with that. So we'll parallelize our process here across two separate threads, which will increase the power or the computational speed. And then once that's all done, we'll save our results using the save command, passing in the results of our growth function, and then specifying where we'd like to save our results to. So in this case, we'll save a zip file of the growth results in our treasure chest. If at any point, if we come back to this and it hasn't completed for you, or if for any reason this cell was aborted, feel free to read in the results from the treasure chest. They already exist there, and it should match exactly what sort of output you would be getting from this. So we'll let this run. All in all, this should take about 20 minutes. But yeah, with that, I think now would maybe be a good time to pick some questions from the Slack, if there's any available.
So there were a lot of questions about model databases, especially when you wanted to model maybe factors from other body types, like vaginal microbiome or oral microbiome, and also like mouse that, like an overview.
Sure. Yeah, so the question was about model databases and what sort of databases might be available for the microbiome, but also for modeling other body sites, perhaps, or in other in the mouse gut, for instance. Off the top of my head, I'm not very familiar with many other databases, but I do know that there are many curated databases available on the web that you could find and pass into MyCom. I think the MyCom data repository has a few databases including the Agora database at a few different summary ranks. So if you were to build the models of the microbiome at sort of the family rank or at other taxonomic ranks, you can pull in the Agora database from the MyCom data repository there.
There's also some questions about the limitations of FBA, and that it can't account for direct toxicity, like direct killing, bacteriocin, or toxicity from tell. It only accounts for metabolic interactions. Talk about that.
Yeah, right. So the question was about the limits of FBA, of flux balance analysis, and whether it's able to account for toxicity of heavy metals and other interactions. And yeah, so flux balance analysis really is limited to sort of metabolic interactions. Doesn't really account for many of those things. For instance, one difficult thing to model is pH in the gut, and that's something that isn't fully captured by flux balance analysis. And there are proxy ways to sort of model these things and include them in the community model. But flux balance analysis itself does have limitations in the sense that it really is limited primarily to metabolic interactions. But that is the case, yeah. All right, well, yeah, I'll jump back into the slides for a little bit here, and then we'll continue. Yeah. So while our communities while our models are building, let's discuss a little bit about, we'll revisit the theme of this year's course, which is, again, modeling the microbiome of historically underrepresented groups within microbiome research. So this visual, which Chris showed us yesterday, shows us just how overrepresented the North American and European microbiomes have been in the field of microbiome research in the past decade. Today, we will build metabolic models of the microbiome for the 3 indigenous groups introduced in yesterday's lesson, which we've already started. And we'll use these models to make predictions of the metabolic behavior of each based on the environmental context that the microbiome is exposed to. We'll also see what work still needs to be done in order to make microbiome research more inclusive of these historically underrepresented groups. And we'll see how current methods can be shaped by overrepresentation of subsets of the population, such as European and North American microbiomes. So as I've already touched on, for instance, the model database that we used to build our models was constructed primarily based on studies of European and North American microbiomes, and therefore is overrepresented by taxa commonly found in microbiomes from those regions. All right, so when I say the environmental context, what do I mean by this? This is the environmental context that the microbiome is exposed to is a key determinant of composition and subsequent metabolic response. So this can take multiple forms. For instance, we have different water sources can impact the composition of the microbiome and subsequent metabolic behavior of the microbiome. Drug use can, for instance, antibiotic use can drastically change the composition of the microbiome and cause large shifts in composition and then ultimately in the growth behavior of the microbiome. Exposure to pets and other animals can introduce new microbes into the microbiome and again, change its composition and therefore its metabolic behavior. And then finally, diet is one of the most important contributors to this environmental context and can drastically change both the composition and the metabolic behavior of the microbiome. So in MyCom, as you've already seen, we can partially represent the environment we can represent the environmental context through the application of an in silico medium that represents the diet available to the microbiome. Since metabolites in the diet are directly feed into internal reactions and are direct precursors of microbially produced metabolites, it's critical that the medium that we use for the growth simulation within MyCom be representative of the diet that's typically consumed by the subject being modeled. If we were to apply a diet that doesn't represent what that individual is actually eating, the predictions that we gain from MyCom will likely be very inaccurate. So today we'll be investigating this by using two separate media for the samples that we've built in MyCom. First, we'll be using a matched medium, which represents the diet of the indigenous groups that we are modeling. And secondly, we'll be using an unmatched medium. In this case, again, we'll be using a representative Austrian diet, which is not typically consumed by the individuals in these three indigenous groups that we're modeling today. Okay, so to use a medium for a growth simulation in MyCom, the medium must be componentized. So what does that mean? That means it needs to be described by its constituent metabolic components. We can't yet say that this person just ate a banana. We rather have to determine what metabolites are in the banana and then add those metabolites to the medium individually. Componentizing media itself is a pretty laborious task, and it requires determining the constituents of various food components present in the diet and then adding them in the proper amounts. For diets of highly represented groups in microbiome research, such as North American or European diets, componentized media are publicly available and easy to find online. However, media representative of the diet of underrepresented groups, like the three indigenous populations we're studying today, is not commonly available and thus needs to be constructed by hand. This can be additionally difficult since the databases that you might use in order to build these media by mapping food components to their constituent metabolites, those databases consist primarily of foods that are common in North American and European diets. For instance, baobab fruit, which is shown in the picture here, is a very common component of the Hadza diet, but it can't be found in any of the commonly used food databases that we use to map food components to their constituent metabolites. The construction of media that's representative of the diet of underrepresented groups rather than the diet of European and North American individuals, as well as the inclusion of food consumed by these groups in these food databases that we're using to build the media, is another issue that must be addressed by the microbiome research community in coming years to make this make research in this field more representative of the world population and more applicable to the world population, rather than just those groups that are historically overrepresented, again being European and North American microbiomes. So for this portion of the course, we decided to construct our own media representative of a standard representative of a standard meal in each of the 3 communities that we are modeling. So we built a componentized representative meal for the Hadza, Chepang, or Mafa communities, each of which were very distinct from each other. First, the first step here was to determine what the meal looked like based on extensive literature review. So for the Hadza community in Tanzania, a representative meal included baobab fruit and seeds, honey, and antelope. For the Chepang community in Nepal, we included nettles, mountain yam, and cowpea. And finally, a representative meal for the Mafa community in Mexico, corn, beans, chayote, and chicken were included in the meal. Each of these is representative of a meal in each of these communities, but is not by any means an exhaustive representation of their entire diet, which would require a much more extensive investigation. Additionally, seasonal dietary variations introduce shifts in metabolic availability to the microbiome that is not captured here. However, each of these diets is a much closer approximation to much closer match to the corresponding community upon whose diet was based than the existing componentized media that we could find online. And the metabolic components in each were quite distinct from the unmatched representative Austrian diet that we're using as our unmatched medium. In order to construct the matched media that we'll be using for modeling the communities, we first had to determine the dietary components present in the standard diet of each community, as I explained in the last slide. So we basically found the food components that are present in a typical meal for each one of these communities. Next, we mapped each food to its constituent components using a food database. Like I mentioned previously, some foods like baobab fruit were not available on these databases. So for instance, in the case of baobab fruit, we found an NMR-based study detailing its metabolic composition, which we could then use to determine those constituent metabolites and their abundance. After determining these constituent metabolites, we add those to our medium in the correct relative abundance. And then following this, we then have to account for adsorption in the small intestine. So many of the metabolic components of our diets are actually absorbed in the small intestine before they even make it to the gut microbiome in order to be consumed there. So we have to account for that in this step. We also add bile acids and mucin, which are introduced along the gastrointestinal tract before reaching the large intestine. And then finally, before we have a completed medium that we can use for growth in MyCom, the medium undergoes a gap filling process, basically augmenting our medium with a minimal set of metabolites that are required for all of the taxa in the sample to be able to grow. So now at this point, we have a componentized medium for each one of our 3 indigenous communities that are being studied. And we can apply this to the models that we're growing. And with cooperative trade-off FBA, we can then use those to return unique growth rates for most of the taxa present in our sample, and then estimate fluxes from those growth growth rates. Excuse me. All right, so I think at this point, our models are likely still growing. So this might be a good point to take a break and then come back to the presentation after the break. And we'll, after the models are finished growing, I encourage you to leave your notebook open to let those complete growing. And then we'll take a look at the output of our results and build some visualizations based on that output. So thank you so much for your attention. And I'll see you back here at believe it's
11:20.
11:20. Great. Thank you so much. Hello, everyone, and welcome back. We'll be continuing the second day of the course now. And to this point, we've learned about flux balance analysis. We've learned about how to extend flux balance analysis into microbial communities using MyCom. We've learned about how MyCom uses that method in order to predict unique growth rates and exchange fluxes for our community models. And we've learned about applying a medium, building and applying a medium to our models in order to model the growth of these communities in MyCom. So at this point, hopefully all of the models have completed growing and are ready for us to explore it a little bit more. But and I know you're probably all excited to see the results of the MyCom growth simulation, and your wait is almost over. But before we jump back to the notebook and take a look at the results of our growth, let's first take a look at some of the visualizations that we'll be able to create with the output from our results, and some of the insights that we can gain from those visualizations. Since MyCom does give us so much awesome data in the output, it can be a little bit overwhelming at first to figure out exactly what we're looking at and what visualizations we can build. But don't worry, it's actually really easy to make great visualizations directly in MyCom or to extract the data to use in your favorite visualization package, whatever that might be. So let's move forward here and just take a look at what some of these visualizations that we can build are. So the first thing that we can look at after we return the results of growing our models in MyCom is visualizing the individual growth rates for taxa in all of our models. MyCom has a built-in function for this, so it's very easy to build using the results of our growth function. Here, we see a scatter plot with the log scaled growth on the y-axis for each taxa that's listed on the x-axis in ascending order of mean growth. This plot, first and foremost, is a great way to check that the models that you've built and then have then grown are growing correctly at a typical growth rate that's, biologically relevant again. And that's sort of what we see here, sort of in that region between 0.001 to 1 is about what we expect to see most of our taxa growing at. If there's a few that are lower, that's okay too, just as long as everything seems to be growing at a rate that we sort of expect. This plot can also tell you a lot about which microbes seem to be doing well in the dietary context that you've applied. And those microbes might have higher growth rates than others. Growth rate is also pushed towards abundance in some MyCom models. So you might see that more abundant taxa seem to have somewhat higher growth rates as well. For instance, here we see Prevotella seems to have a very high growth rate in all the communities in this set of models. And this tells us that Prevotella seems to do very well in this dietary context that we're using to grow the models here. We'll also see in our results, since we are modeling our communities on 2 different diets, that the growth rates will change somewhat between the 2 dietary contexts, which makes sense since the dietary context changes, the metabolic behavior of imports and exports changes. Therefore, we expect that the growth rates might be a little bit different. This echoes one of the primary points that we're making here today, which is that diet is a major factor in the metabolic and growth activity of the microbiome. And it is important to choose when you're doing metabolic modeling of a community. It's important to choose a diet that matches the diet that's actually consumed by the individual who you are modeling. So choosing a representative diet is a critical step in metabolic modeling. So beyond looking just at growth rates, we can make a few other visualizations as well that we'll take a look at in the notebook shortly. So another thing we can look at is what's called the niche space. So if you're not familiar with niche space, this is basically the niche space is a measure of the context-dependent way in which the taxon uses its environment. So in this case, we're measuring how the metabolites that each taxon imports is different. Since plotting all of these individual import reactions wouldn't be possible by itself, we'll use a t-SNE plot to make this visualization. So this is a technique that takes high dimensional data, for instance, all of the imports into each of our individual taxa, and reduces the dimension down into a two-dimensional plot like the one that we see here. And it builds this plot in such a way that similar points are closer together and less similar points are spaced a little bit farther apart. So points on here that we see very close together are typically we can infer that this means that they're importing similar metabolites as others that are close to them. And 2 points that are farther away are importing very different sets of metabolites. And we can call sort of each point on this map, or each section of this map as sort of a different niche. So like I mentioned previously, niche space is context dependent. So what that means is that depending on the dietary context that we're using to build our models, the niche space might change significantly. So some taxa might change their metabolic behavior depending on what metabolites are available and present in their environment. And this can allow them to survive in multiple niche spaces. So this is an adaptation that can be helpful for their survival, and one that we see commonly in the bacterial kingdom. What we'll see when we're comparing the relative positions of taxa between the two dietary contexts we're using, the matched and the unmatched diets, is that many will change their predicted metabolic behavior. And because of that, they'll actually occupy different niche spaces on this two-dimensional t-SNE plot that we're looking at here. So we might have two taxa that are very close to each other in the unmatched dietary context. But when we switch to the matched dietary context, the one that actually represents the diet of the individuals for whom we're modeling, those points might move farther away, indicating that they're now importing different metabolites than they were in the unmatched dietary context. This again just underscores the need for accurate matched dietary media when we're building these metabolic models, as if we use, due to the fact that if we use an unmatched medium, we'll end up with predictions that differ quite drastically from what might actually be happening in the environment that we're modeling. So this is one plot that we can use to learn a lot. But let's move on and look at the last plot that we'll be taking a look at. So here, what we're looking at is what we call comparative metabolomics. So in this case, what we're looking at are metabolic exports predicted from the growth of each metabolic model. So as with the one that's shown here, what we'll do in our notebook is construct a heat map that illustrates the amount of different metabolites that are produced and then subsequently exported from each taxa, from all the taxa in each sample. So in this case, in the case of this heat map, each row represents the export of an individual metabolite, which are annotated on that y-axis. Each column represents a distinct sample in our set of samples that we're modeling. And the cells are colored in with a logarithmic color map. So basically, we're showing the logarithmic flux out of each one of these samples of each given metabolite. Microbiome has a built-in tool that we could use to build heat maps like the one shown here very easily. But we'll actually use this opportunity to take our output from our growth command and build a visualization in another package on our own. In this case, we'll be using Seaborn, which is a common package in Python used for building visualizations. Additionally, this will allow us to plot the growth of both diets on both dietary contexts on a single plot. We'll be able to compare the metabolic output of our samples on each dietary context on the same heat map. So similar to what we were talking about with the niche space, the metabolic activity of any taxon in a community, and therefore also its exchanges, is dependent on the context in which it grows. So in this case, again, we're looking at two dietary contexts, one matched, one unmatched. And since the environmental context in these two simulations is very disparate, we expect to see very different exchange fluxes occurring between each of these two dietary contexts. And I probably sound like a broken record at this point, but this again just shows that it's very important for us to consider the diet when we're building these metabolic models and simulating their growth. Since the dietary input feeds directly into the production of these metabolites that are subsequently exported from the microbiome, it's very important that we get the composition of the medium correct and make it representative of the models that we're growing. Cool. So after we build our visualizations, we'll actually pass it off to you, and it'll be your turn to jump in the driver's seat and explore using MyCom a little bit on your own. So after we build our visualizations, you'll find some cells at the end of the notebook that you can use to explore loading a single model, and rather than building multiple models at once. And from there, applying a medium like the ones we were using today and simulating growth. You'll then have the opportunity to kind of explore on your own, whether that be measuring outputs and exchanges or building your own visualizations, either directly within MyCom or in your own, another Python package that you might use for visualization. So you get to look forward to that. But first, we'll switch back over to our results in our notebook and build some visualizations and take a look at our output of our growth command, and then get into building some visualizations that will give us further insight into the results of our modeling that we've done here today. So let's all switch over back to the notebook and take a look. So again, up to this point, what we've done is import our CHIME artifacts that we built yesterday, which include our abundance and taxonomic assignment. We've used those along with a database of genome-scale metabolic models to build our metagenome-scale metabolic models using the MyCom grow command. We've then loaded in our medium, which again is a matched medium representative of a typical meal in each of the three indigenous populations that we're studying. And then now we've gone ahead and we've grown those models on that matched medium. And hopefully at this point, all of your models have completed growing. And we have output from that should have been saved into our treasure chest. If for any reason it's still going, or perhaps it was aborted during the break, feel free to run this next cell, which will load in the results which have been pre-saved in the treasure chest. That'll allow you to run the next cells, and the growth should be identical to the output you would have gotten from this cell here. Great. So what kind of results do we get from this MyCom grow workflow? This grow command returns a tuple of three different data sets. So a tuple is a data type in MyCom, and within that tuple, we have three separate data frames. First, we have a data frame that shares with us the predicted growth rate for all the taxa in all the samples. So basically, this uses that cooperative trade-off flux balance analysis method that we discussed previously in order to predict unique growth rates for most of the taxa present in our samples. And we'll take a look at that shortly to make sure that everything went the way that we expect it to. Secondly, we also are returned a data frame showing us the import and export fluxes or exchanges from each taxon and to the internal environment, external environment. This data frame will be very helpful to us when we're building our niche space visualizations and our comparative metabolomics visualizations. And this will tell us sort of more about the differential metabolic behavior of our metabolic models between the two dietary contexts that we're studying today. Finally, the grow command also gives us a data frame of annotations, which correspond with all the fluxes that have been predicted from the metabolic models. And these annotations just tell us what metabolites these fluxes correspond to, as well as mapping them to a few other databases that you might be using for research purposes, such as KEGG or HMDB. Great, so let's take a look now at this first result from our growth command, which is our growth rates data frame. You can very simply pull this up by calling the output of our growth command, which was matched underscore growth, and then following that with growth rates. So we'll run this cell and take a look at the output. So this is pretty straightforward. We have each one of these rows represents a single taxon within a single sample. And in here, we can see the abundance of each taxa within that sample, given in terms of relative abundance here. We also see the estimated growth rate predicted by MyCom using that cooperative trade-off FBA scheme that we discussed earlier. See the number of reactions and metabolites given in that the reconstruction of that taxon. And then we can also see the sample ID in which this taxon resides. So this is a good way to take a look at the results of your growth command and make sure that everything is, all the taxa present in the sample are able to grow at a rate that you might expect to come out of this. So everything here looks pretty good. We see all of these growth rates are sort of in a range that we expect and is appreciable. And so this is pretty straightforward, and it looks like everything has worked correctly. So that's great. If you wanted to calculate the overall community growth rate within a single sample, you could multiply the growth rate column by the abundance column, and then sum together the resulting product across all the taxa within a sample. And that would give you the overall community growth rate, like we discussed earlier. The next output from our MyCom grow command is the exchanges data frame. So this is a little bit more interesting in my opinion. And this data frame will show us all of the import and export fluxes from the system into the external environment. So let's take a look at this data frame now. Again, we can just call the result of our growth command and then follow that with .exchanges. Cool. So in this data frame, it's a little bit longer, and that's because each row represents a single metabolite within a single taxon within a single sample. So that's why we end up with a data frame that's nearly 10,000 rows long. You'll see that one of the taxa included as well is the medium, and that's because the medium in the MyCom model has its own compartment that can import and export taxa, as with import and export metabolites as with the other taxonomic compartments. So what we see here is the sample ID in which the exchange is happening, the name of the reaction given by the name of the reaction corresponding to the export flux of a metabolite, and then the annotation of the metabolite itself over here. The flux column tells us the either import or export flux of that metabolite into or out of the taxonomic compartment that we're looking at. A positive flux indicates export, and a negative flux indicates import. And we can also see that in this direction column on the right-hand side. So for instance, here, we see that our sample, this sample given here, is exporting this metabolite at a rate of 0.0036 millimoles per gram dry cell weight per hour. It's important to note that all of the fluxes shown here are normalized to 1 gram of dry cell weight of bacteria. And this is done so you can directly compare fluxes between taxa, even if they are present at very different abundances. It's important to note, though, if we want absolute production of each one of these metabolites and absolute exchange, we would need to multiply the predicted flux by the abundance of that taxon in order to determine the metabolic contribution of that metabolite by that bacteria. If we want overall production rate from an individual sample, we need to repeat that across all of the taxa present in that sample. Luckily for us, there is a built-in function in MyCom that we'll discuss shortly that will do that math for us and will return to us absolute production rates from each sample of all the metabolites listed here. Now, the final result from our MyCom growth function is the annotations data frame. And this is useful if we want to learn more about any of the exchanges that we see in the exchanges data frame. So if, for instance, we see a metabolite that's designated BUT, like this one here, we can go to the annotations data frame, we can search for that metabolite, and we can learn a little bit more about it. Cool, so it looks like the metabolite designated BUT is butyrate, which is a common short chain fatty acid produced by the microbiome. That's actually a pretty important metabolite. You can follow this link if you want to learn a little bit more about butyrate and its role in the microbiome and exchange with the host. And we also see in this annotations data frame a couple facts about butyrate and the exchange of butyrate. So we have its molecular weight, the number of carbons, number of nitrogens, and then butyrate's annotation in a few different databases as well. So HMDB, KEGG, PubChem, and so on. We also see the corresponding exchange reaction for butyrate, which is this reaction shown here. All of these annotations and also more information about them are available at vmh.life, which is a database maintained by Dr. Ines Thiele's lab that contains all of these metabolites as well as involved reactions and which reconstructions these reactions take place in. All right, now last thing before we get into building our visualizations ourselves was we wanted to directly compare the results that we gained from building our matched medium against what we saw. We wanted to compare the results we got from growing on our matched medium against results we obtained from growing these models on an unmatched medium. In order to illustrate just how important it is to use proper dietary inputs for the modeling process. Again, it's really important to match the dietary input to the model that you're growing. So rather than repeat all the steps above, so regrowing all of our models on a different medium, we'll just load in the results from the treasure chest that are pre-saved there. So the following cell will load in the growth results for the same models as above, but using an unmatched medium. And we just did this in order to save you a little bit of time, since that would take about another 20 minutes in order to grow on a second medium. So we'll just run this cell, and then we'll have growth results available for both of our dietary contexts, namely matched growth and unmatched growth. And if you are interested, you could also take a look at the growth rates and the exchanges data frames from the results of this as well, from the unmatched growth results. Cool. So now we can get into building visualizations of our output. Visualizations allow us to interpret these results in a more meaningful way than just looking at these data frames. And like I mentioned in the slides, it can be a little overwhelming, but there are tools in place already to build these visualizations that can give us a little bit more insight about the models that we're building and growing here today. So first, we'll take a look at some standard visualizations included in MyCom. These tools just take in the growth results data frame that we just took a look at and creates visualizations directly and save those as HTML files that are output into our materials folder in our file explorer on the left-hand side. So what we'll do here is we'll import our MyCom viz module. So what this does is it imports all of our visualization tools given in MyCom, and then we'll run the plot growth command. So what this does is it builds the plot that we saw in the slides, showing the individual growth rates of all taxa in all our samples. We'll pass in each one of our dietary context growth results, so unmatched growth and matched growth. We'll also designate a file name. So then the output of this cell will save these HTML files into our materials folder on the left-hand side. The cell runs pretty quickly, and these should pop up. If you don't see them pop up, you can click this refresh button, the second button from the left under the Files tab, and we should see them pop right up. So here we have our matched growth rates.html and our unmatched growth rates.html. We can't open these directly in our Google Colab notebook, but what we can do is download these each individually and then open them directly in our browser. So let's go ahead and do that. First the unmatched and then the matched growth rates. You may have to resize the page a little bit in order to see the full, in order to see what we're looking at here, which again is this similar plot to the one that we saw in the slides. And so what we can see here is that is again on the y-axis the log-scaled growth rate of all the taxa present in our sample, which are again listed on the x-axis. And you'll see that there's a fairly similar shape to the overall data, but there's definitely significant differences in growth rate between the two dietary contexts for all of our samples. For instance, if we take a look on the far right-hand side, in our unmatched growth rates, we have these blue dots representing Prevotella. If we take a look at the results shown in the matched growth rates, we actually see that Prevotella, the growth rate of Prevotella seems to jump quite a bit higher in our matched growth rates versus the unmatched growth rates. This makes sense since the matched dietary context that we used actually includes a lot more fiber than the unmatched growth rate. Excuse me, the unmatched diet that we applied to the unmatched growth results. And Prevotella is known to thrive in dietary contexts in which there's a lot of fiber. We see a little bit of a difference further along the line here too. Like I mentioned in the slides, this graph is ordered in terms of sort of increasing mean growth rate. And so we can see that the order of taxa along the x-axis shifts a little bit between the two dietary contexts. And that's because some taxa are able to grow at higher growth rates and some are growing at lower growth rates between each of these two diets. I encourage you to take a look at some of the other taxa and variations in between the two dietary contexts. A cool thing about this tool is when we build this visualization, if we select any of the points, it will show us all the points within a single sample within that plot. So that's an easy way to compare differential growth rates for a single sample between the two dietary contexts that we're looking at here today. So again, if you're interested in looking at this a little bit more, definitely take some time to browse through these plots we've built and take a look at how the growth rates can change between a matched and an unmatched dietary context. Great, so now we've built those. Let's return to the notebook briefly and take a look at the next visualization that we'd like to build here. So as I alluded to in the presentation, another thing we're interested in looking at is growth niches and how growth niches might change between the two dietary contexts we are studying today. So here we can use the plot exchanges per taxon function, which again is a built-in MyCom function, to which we can pass in our growth results, and that will construct a t-SNE plot of niche spaces, like the one that we saw in the presentation slides. So again, we'll run this plot exchanges per taxon function, pass in the results of our growth function, so our unmatched growth and our matched growth. Perplexity is a t-SNE parameter that you can change in order to get more meaningful groupings. And if you're interested in this, I encourage you to play around with some of the parameters and see how that changes the results. For now, we'll stick with 4. We'll also define which direction we're actually looking at the niche space from. So in this case, we're interested in niche spaces which are defined by the metabolic imports of each taxon. Basically, what metabolites in the environment is the taxon eating in order to grow. And how does that change between the two dietary contexts? You know, if you switch to a more high fiber diet, for instance, is each taxon suddenly switching from eating more sugars to eating more fibers? So we'll define the direction to be import. So we're looking only at import fluxes. And again, we'll name our files so we know where to find them in our file explorer. We run this cell. MyCom will go ahead and build these visualizations for us and output them to the file explorer on the left-hand side. Again, if you don't see them, go ahead and hit that refresh button and they should pop up like so. As with before, we'll download these files individually and then open them in our directly in our browser. And that'll be the easiest way for us to find easiest way for us to visualize these. So the matched one first, and then the unmatched growth. And again, you might have to resize your browser window a little bit in order to see the full plot. Great. So as I mentioned previously, the t-SNE plot, what this does is it takes high dimensional data. So for instance, all of the import fluxes into each one of these taxa, reduces it down into 2 dimensions. So into two-dimensional data that we can plot like the plot you see in front of you now. It does this in a way that points close together are more similar to each other. So in this case, they have similar metabolic imports, and points that are farther apart will have very different metabolic imports. One way to look at this too is that anything that's closer to the center of the plot, for instance, will be ostensibly competing with the most different taxa for their niche space. So if we take a look here, we can see sort of 2 clusters of taxa, but we also see some very distinct regions and points that are fairly far apart, indicating that their metabolic imports are very distinct from each other. We can take a look at the unmatched dietary context, and we see a bit of a different shape. Still maybe 2 overall niches, but some of the taxa have moved around a little bit. And so what's interesting about this plot too is that we can select individual points, and it'll show us all the taxa across all this, or it'll show all it'll show that taxa across all of our different samples, is what I should say. So for instance, here we see Clostridia, and we see all the indications of Clostridia across all the samples that we are modeling today. So for instance, here we have 5 Clostridia that are pretty close together here, and then 1 that's way over on this side, indicating has very different metabolic imports than the others. If we look at Clostridia on this plot instead, we'll see that they're a little bit more spaced apart, and rather than having 1 that's farther apart, we actually have 2 that are more on this side of the plot, while 4 are on this side. Another interesting one that I'd like to call out is Blauschia. Over here, we seem to have 2 sort of individual niches for Blauschia. Again, this is on the matched diet. So we have 2 sets of Blauschia that seem to have very similar metabolic imports when compared to each other. If we take a look at the matched dietary, at the unmatched dietary niche, excuse me, we actually, oops, that's the wrong one. I'll have to find Blauschia, there it is. What we see is that we actually have still 2 niches, but it seems that one of them has now sort of switched its niche closer to these 2 rather than where it was closer to this one earlier. So what we see here is, and I encourage you to take a look at these plots a little bit more and see what other interesting patterns you can find. Maybe you can share them in the Slack channel. But really what this shows us is that when we switch from one dietary context to another dietary context, some of these taxa are behaving very differently, metabolically speaking. They might be, in this case, importing different metabolites that they use in order to grow. And so, again, this shows us just the importance of diet in this modeling process, and why it's important to figure out exactly what sort of metabolites are actually available to the microbiome in order to make our predictions more relevant to what's actually happening. And that gives us more clinical relevance, really. Cool. So again, I encourage you to take some time to look at these plots, see what other patterns you can find. But in the meantime, we'll switch back over quickly to the course notebook. And from there, we'll build our sort of last visualization, which again was a heat map that will give us an idea of sort of comparisons of metabolic exchange from each one of our samples. Metabolic export, I should say, from each one of our samples. So yeah, we're comparing the metabolomic export from each of the 2 dietary contexts, and we're interested to see how that profile shifts when the diet changes. So can we sort of visualize how different metabolites are exported differentially when grown on a matched diet and on an unmatched diet? And so to do this, we'll actually take our data out of MyCom, and we will build this visualization in Seaborn, which is a common visualization platform in Python. But first, we'll have to manipulate the data a little bit. And to do this, we'll be using mainly pandas. If you're familiar with pandas, this should be straightforward. If not, then this will be a good introduction. And feel free to look at this notebook a little bit longer if necessary. Great. So the first thing that we'll do is we'll import this production rates function from our mycom measures module. So as I alluded to earlier, the exchanges data frame that comes out of our growth function gives us exchanges normalized to 1 gram dry cell weight for each taxon. So then we can compare exchanges between taxa directly. But since taxa are present in different abundances in a sample, we have to account for that when we're calculating production rates. So what this production rates function will do is it will multiply those fluxes by the relative abundance of the taxon from which they're originating. And then it will sum together all of those production rates across those taxa within a sample. So then what we're left with is the overall production rate, the overall export flux of a metabolite from a single metabolic sample, single metabolic model. So this is a nice, easy way to arrive at that result without having to do all the math ourselves. So what we'll do here is we will pass in our growth results into this production rates function provided by mycom. And then we'll also add in a column that we'll name diet. And in this case, we'll make all the entries in that column matched. We'll do this because later on, we'll concatenate our two data frames together. And we'll want to know which dietary context each of these production rates comes from. So we'll then do the same with our unmatched production, with our unmatched growth results. We'll put those into the production rates function, and then add a column into the resulting data frame that we'll call diet, and we will fill that in with unmatched. So then, as I just mentioned, we'll concatenate those two production rate data frames together. And we'll do this so that we have all of our data in one place. The seaborne heat map that we'll be constructing is able to read this data frame directly, and therefore we can, we should be able to, if we concatenate them together, it'll be much more easy to build one singular visualization that we can use to compare metabolic exports directly. So like I mentioned, Seaborn can read this data frame directly, but what it wants is basically a pivoted table that looks like the heat map that I showed you in the slides earlier, wherein each column represents a sample and each row represents a metabolite. So what we'll do is we'll use the pandas pivot table function, and we'll pass in our exchanges data frame, which is again the concatenated data frame of our production rates on both the matched diet and the unmatched diet. We'll then set the index to be both diet and sample ID, since each sample ID is repeated twice for each dietary context. So this index will then organize those by both diet and sample ID. We'll pass in, for columns, we'll pass in name, which in this case is just the name of the metabolite that's being produced. And then the values within each cell of the data frame will correspond to the flux of that metabolite out of that individual sample within the dietary context. If any of this is hard for you to visualize just by looking at the code, I encourage you to run each section of this cell individually and take a look at the output to fully understand what the different commands in this cell are accomplishing. And then just a few more things we need to do in order to clean up the data so we can use it for our heat map. Our data frame at this point will include some cells for which we don't have values. And that's because mycom might be predicting that a given sample may not be producing one of the metabolites present in our data frame. So for any samples for which we don't have values, we'll fill in those cells with zeros, with the assumption that if it's not predicted by mycom, that indicates a flux of 0 out of that sample. Finally, what we'll have to do is a center log ratio transformation again. This is the same as what Christian did when he was plotting taxonomic abundances yesterday at the end of yesterday's course. So basically just taking the log value of each cell and subtracting the log mean from that. We do this so that we can compare fluxes directly, even if they are orders of magnitude apart. Finally, we'll just reindex our data frame and sort the data frame based on variance, just so that we have samples with the highest variance at the top. And this will just make plotting it a little bit more interesting. So if we run this cell, we can take a look at our output, which we are calling our exchanges data frame. And again, so what we see is a concatenated data frame showing the center log ratio transformed flux for each one of these metabolites out of each one of these samples between our two dietary contexts. So we can now pass this data frame directly into Seaborn, which will read each one of these cells and construct a heat map that's shaped similarly to what we see here. Cool. So like I said, we'll use Seaborn to plot our heat map. And in this case, we'll actually use a cluster map, which will cluster together similar rows. So samples that are exporting metabolites in a similar manner. We'll pass in just the first 50 rows of our exchanges data frame. Otherwise, this plot would be far too long. As you can see, this data frame is 628 rows long. And we'll define the color map and the figure size that we're using. Cool. So what we can see here is the resulting cluster map. And I encourage you to, again, take a look at this and see what patterns you can find. But overall, what we can see is that the dietary context that's applied to each one of these samples is important. There's significant differences in export fluxes between the matched and the unmatched diets. Overall, we can also see that there's sort of clustering of unmatched and matched samples together, indicating that there's clustering of unmatched samples with other unmatched samples and matched samples with other matched samples, indicating that they act similarly when given the samples within the same indigenous community act similarly when given the correct diet. And there are more defined patterns that we see when we're looking at the matched diet versus sort of the unmatched diet. So again, this really, and I'll say it one more time, and then I'll be done, I promise. But this just underscores the need for correctly matched diets with the metabolic models that you're building. We can see that there's really different expected results from these MyCom models based on which diet we're applying to these metabolic models. So you can imagine if we're applying the wrong diet and we're getting very different results, we might be led to draw different conclusions than if we apply the correct diet that matches the models that we're trying to build here. Cool. So as I mentioned before, now that we've sort of built all our visualizations and you've learned the basics of MyCom, going from building the models to growing the models to visualizing the outputs, now it's your turn to kind of jump in the driver's seat and take a look at some of these diet-specific interactions that we've been discussing here today. This first cell will guide you through how to load in one individual model and grow models individually rather than growing them all at once. And after that, you have space to go ahead and try growing models on other available mediums, as well as trying your hand at building your own visualizations and finding some insights from that. Additionally, at the end here is a piece on the workflow that we use in order to select a trade-off value. If you're interested in using MyCom further for your own research purposes, I encourage you to take a look at this and learn a little bit more about how you might choose a trade-off value when simulating the growth of these models. So that's all for you to do at your own leisure. And if you have any questions, feel free to ask on the Slack channel. So with that, we're done with the second part of the ISB, the second day of the ISB microbiome course for this year. Before we end, I want to sincerely thank everyone that was involved with putting this event together. You've seen my face and Christian's face for the last 2 days up here presenting, but none of this would have been possible without cooperation by everyone that was involved in putting this together. A lot of people working behind the scenes to make this happen, and I am sincerely thankful to all of them for making this happen so that I could be up here and talk to you guys all day about metabolic modeling and some of these cool methods that we're learning about here. I also want to thank our sponsors for letting us put on this event for free to try to spread this information to as many people as possible. And last but not least, I want to thank all of you for being here today and sharing your excitement and enthusiasm about microbiome research with all of us and allowing us to spend the day teaching you about some of these cool things that we're learning in this rapidly developing field. I hope you learned something and I hope you enjoyed yourself. And again, if you have any questions or comments, please just mention them on Slack and we'll be happy to get to you just as soon as we can. We'll be taking a break here for a little bit, but next up. Oh yeah, sure, absolutely. If there's any. Yeah, sure. So the question was about how we validate some of these predictions that we make in MyComp. So as I mentioned previously, the growth rates that are predicted by MyComp have been experimentally validated by looking at metagenomes. And from metagenomes, we can estimate growth rates based on coverage profile, or sort of decay of coverage profile away from the origin of replication, which lets us calculate sort of an estimated growth rate for taxa. We can then compare that against the growth rates that are predicted by MyComp, and we see that actually with cooperative trade-off flux balance analysis, we end up with growth rates that are well correlated with estimated growth rates in taxa in vitro. When it comes to validating exchange fluxes out of our metabolic models, one approach that we've looked into is using ex vivo models for measuring sort of metabolic flux out of a community and comparing that to the metabolic flux predicted by MyComp. And we found that actually with certain parameters, we are able to replicate ex vivo exchange fluxes from communities using MyComp. So that validation work is ongoing, but it does seem to be that MyComp can capture some of these metabolic fluxes very accurately. Any other questions?
You just say
12:45. Yeah, so we'll take a little bit of a break now, and at 12:45 we'll have the very talented and distinguished Alex Carr come up here and go say talk about some of his research that's he's been doing here at ISB, focusing on microbial ecology in the microbiome. So please join us back here at 40 minutes at 12:45 PST. Hello, everyone, and welcome back to day 2 of the ISB Microbiome Symposium, microbiome course. In a few minutes, we'll hear a talk by Alex Carr. Alex is a senior PhD candidate in the molecular engineering department at the University of Washington in Seattle, who is conducting his research here at ISB. Alex received his undergraduate degree in chemistry from the University of California, San Diego. Upon completion of his undergrad degree, Alex joined the lab of Adam Arkin at Berkeley, where he worked on characterizing and modeling dynamics in synthetic consortia of human gut microbes. Now at ISB, Alex is co-advised by Drs. Gibbons and Baliga and conducts research focusing on furthering our understanding of how interspecies interactions and environmental factors facilitate the formation and functions of microbial communities, how these communities adapt to changes in their environment, and the functional roles they play in the soil and human gut ecosystems. So without any further ado, I'd like to welcome Alex and take it away.
Thank you, Nick, for that lovely introduction. Okay, let's see, I'll share my screen. Oh, it's already shared. Great. Oh, let's see, share. There we go. All right. So yeah, thank you everybody for coming back for day 2 of the microbiome course. And yeah, let's get right into it. So I'll be telling you about some of the research I've been doing in the Gibbons lab, leveraging community level metabolic models to predict C. diff colonization. So yeah, why do we care about C. diff? Well, it's actually one of the leading causes of GI tract infections. Here in the US, at least, C. diff infections are a serious issue which affect nearly 500,000 people annually, 6% of which are fatal. And while the rate of infection has been decreasing in recent years, it's still a serious burden. And we know, based on the research that's been done so far, that factors such as antibiotic use, diarrhea, and other disruptions to the host microbiota are major risk factors for infection. And some of the progress thus far, we know, for instance, that fecal microbiome transplants are very effective remedies, and that's because they are able to reintroduce healthy microbiota into the community that can outcompete C. diff for the niche it occupies. And through correlative studies, we know that there are numerous gut microbes that have been shown to be associated with disease prevention. Excuse me. But there's an important distinction here between colonization and the transition to infection. One thing, for instance, is the fact that more of us actually have C. diff than you might expect. In adults, there's estimates put it around 4 to 15% are asymptomatic carriers here in the US. And what that means is that you can be colonized by C. difficile, but not necessarily have the symptoms of a C. difficile infection or really any negative consequence. And this is primarily facilitated through acquisition of spores in the environment and other factors. If we look at infants, the rate of colonization by C. diff is actually a lot higher. Some estimates put it around 30%, and with no real detrimental effect from its presence. And so how does infection develop post-colonization? Well, again, this is really due to disruptions in the microbiome, which open up a niche space for C. difficile to thrive. And the way this one mechanism by which this can occur is the fact that C. diff is a spore-forming microbe. And so these spores allow it to persist following disruptions. There are also antibiotic resistant strains of C. diff that have been found in the clinical settings. And so when C. diff is able to persist through these disruptions, whether it be diarrhea or antibiotic use, it really kind of just opens up this niche and without any competition, allows it to bloom, leading to CDI. And so really to kind of address this question, ourselves and many others in the research community have sought to try to understand the interactions in the microbial community that can prevent and promote colonization, so that ideally we can move past FMT and really be able to design our probiotics that might be able to aid the prevention of this disease. And so here I'm showing you just kind of a schematic for the kind of workflow that I think is just kind of a general principle that we use in gut microbiome research. You know, we want to understand the community, so we look at the DNA, whether it be 16S or metagenomics, and then through this analysis, we can get an idea of the composition of the community, whether it be over time or cross-sectionally across populations, and ultimately, at least in this case, we want to really understand the kind of interactions that might be putative interactions that might be occurring in the community. And generally, you know, we will use like correlative approaches to look at associations, but we know this approach is fraught with challenges. And so the research that I'm doing here in the Gibbons lab is seeking to supplement this data with metabolic modeling techniques so that we might get closer to understanding the mechanistic underpinnings of these associations and interactions. And so to do that, I'm using a mechanistic modeling framework, this MyCom, which you've heard a lot about today. And so, yeah, I'll just show you how I'm using this tool to understand how C. difficile is colonizing the gut microbiome. And so, important distinction here is because in the metabolic models, we don't really have kind of an integration of the host side, and really like not a great representation of the formation of the toxins that C. diff produces. So really, we're just kind of looking at colonization, which is the first step, as I mentioned, in the development of CDI. And so here's just an illustration of the methodology that I've used to try to dissect this phenomena. The basic idea is to try to identify resistant and susceptible microbiota. And we do this by challenging, we develop these metabolic models, and then we challenge them by introducing C. difficile, a genus level Clostridoides difficile model into the microbiome at a fixed percentage. And then we ask the question, does it grow? And we use MyCom to answer this question. And in the microbiomes where we do see appreciable predicted growth, we can then use the predicted fluxes to understand what niche the C. difficile might be occupying, what strategies it's using, and how the community may or may not be facilitating its colonization. And so we started this using some publicly available data, time series that were generated in the Eric Alm's lab, where here I'm showing you a time series for an individual over a period of close to a year. And what we see here at the top is their microbiome profile. And here, where I've indicated this arrow, where you see this large bloom of proteobacteria, this individual actually got food poisoning, which facilitated a diarrheal event. And what you can see down below in the trace, in the blue, what we see is this person prior to this event did not have C. difficile or C. diff in their system, but following this event, you see they appear to be colonized by C. diff. And you'll notice too, like the trace is pretty spiky and the relative abundance is quite low. And this is because, we're pretty close to the limit of detection here. So you might expect this kind of spiky behavior. And so then I took this data and essentially did this in silico invasion assay that I just mentioned on the previous slide. And what we see is the predicted growth rate from MyCom. It shows fairly good qualitative agreement with the empirical result. We see prior to the food poisoning event, the predicted growth rate of C. diff is negligible. It's 0. But following the event, you see the predicted growth rate exhibits the same or very similar spiky behavior as the empirical abundance. And so the model would predict that this individual is susceptible, and empirically we see that's true. This data set also provided another example, another individual who similarly was microbiome was followed over time. And what we see in this individual is that actually, again, they had an event that led to diarrhea. But in this particular case, it appears they were colonized by C. difficile even before that event. And you can see it was detected several times throughout the study. And here, MyCom again is showing similar behavior in that it suggests this person is susceptible at different time points and throughout the duration of the study. So again, we see a good qualitative agreement. And so this initial result was promising and motivated us to kind of look further and see if we could better validate these predictions. So to do that, we pulled down some additional publicly available data, this time for people receiving fecal microbiome transplant. So the DIS data set provided data on individuals who came in with gut dysbiosis, and they received an FMT, and in the majority of the cases, the recovery was observed. And so what we see here in this violin plot is that prior to FMT, the predicted growth rate, the log growth rate of C. diff in the models is fairly uniformly high. But then post FMT and upon recovery, we see while there is still an appreciable fraction that are predicted to be susceptible, you see more of this bimodal behavior where there's a much larger fraction which are predicted to be not susceptible to colonization. And so looking at the diversity of the microbiomes in these groups, we see that as expected, prior to FMT, the individuals coming into the clinic had low Shannon diversity, indicating dysbiosis in their guts. And post FMT, we see that the diversity largely recovers, except for in a few cases. And so if we look, these are for the individuals and their traces. If we look at the community import fluxes and the trajectories that these individuals followed, one of the things we see is that here in these EU maps, what I'm showing you are the community import fluxes, so what the community is predicted to consume in the absence of the C. diff challenge, and then colored by the predicted growth rate of C. diff when it is introduced. And what we see is this clustering behavior where you see the community import flux or what it's consuming does seem to do a decent job of predicting whether or not C. diff can invade. And then on top of that, what I'm showing you in these 4 different plots are 4 different individuals who came in starting at the red point, the red circle, where you see in just about every case, they are starting at a point where the predicted growth rate is high. And then the individual traces are the trajectories through this predicted space. And you see in the vast majority of cases where you have the red star, they end in a state where the predicted growth rate is low, but there is a lot of bouncing back and forth in some cases, between high predicted growth rate and low predicted growth rate, which might be suggesting that while many of these individuals recovered, there was always potential for a second reoccurrence. And we know this is a fairly common issue for CDI. And so kind of digging into this a little bit more, these results were suggesting that the community really plays an important role in whether or not C. diff might be able to colonize. And so we sought to understand whether it is capable of occupying multiple niches. And the answer to that is yes, it does appear to have multiple strategies. So here, what I'm showing you is the same FMT data set. Now it's focused on just the import fluxes of C. diff. And so each row is a metabolite flux on the log scale. And you see we're clustering both on the metabolites, but also on the patients. And then at the top, the red bar is the growth quantile for the predicted growth rate of C. diff. And what we can see in this heat map is there appears to be at least 3, maybe 4 distinct clusters where you see there's kind of differences in the pattern of import fluxes. For instance, on the far left, we see this quantile where the growth is relatively high. You see high import of many of these metabolites, several of which are known to promote C. diff's growth in vivo. So for example, we see succinate, ornithine, and trehalose, which are all known to promote C. diff's growth. But an interesting feature here is that we see in the far right, there's C. diff's also able to obtain fairly high growth rates, but using only a subset of the metabolites present. And so again, this is suggesting that it might have multiple strategies for colonization. And so to see whether or not this is specific to this data set or whether it generalizes across data sets, we pulled down some additional data. So Burkle et al. Is the FMT study. David et al. Is the time series that I showed you previously. And then supplementing those two, we also have the Airville data set, which is a fairly large, densely sampled cohort that was generated through a study here at ISB, as well as the American Gut Project, which is actually now the MicroSETA initiative. Initially, this was a North America specific endeavor, but has now sought to kind of globalize their effort. And so what we see here, again, these are the import fluxes for C. diff specifically that have been reduced to 2 dimensions using UMAP. What we see is across these 4 very different data sets, you see very similar structure in the shape of this import flux space. And what this really is suggesting is that, at least in this modeling framework, the strategies used by C. diff appear to be very similar in these different settings. And one of the major differences really just being the sparsity of the data, where you see the larger data sets have the pattern is much more filled in. And so this is very promising results. And so to dig into this just a little bit further, we sought to understand whether what are some of the relationships between the predicted growth rate and some of the, like for instance, the Shannon diversity, which we know is a predictor of dysbiosis in the gut. And one of the interesting things that came out of this, here I'm showing you the relationship between the growth rate, predicted growth rate in Shannon across all the data sets integrated. And one of the interesting things here is, while initially you might expect a kind of negative linear relationship, as the Shannon diversity increases, maybe you'd expect the log growth rates to decrease, until it's at its lowest at the highest diversity. But what we actually see is this sort of sweet spot where you see there's a kind of global minimum here at an intermediate Shannon levels, but the growth rate increases again as the diversity becomes very high. And while this might be surprising, it actually does kind of align with some of our empirical knowledge about diversity in the gut microbiome. We know when people have diarrhea or there's dysbiosis, the diversity tends to be very low. But we also know in other settings, such as when individuals are constipated, the diversity can actually be quite high, but this isn't always necessarily a good thing. And so one mechanism here might be that in the higher diversity settings, there's potentially more individuals that might be creating niche space for C. diff. And so to try to understand that, we looked at the community itself to try to identify which microbes might be competing with C. diff or promoting its colonization. So to do that, one kind of initial pass through the data then identify some putative hits was to simply correlate the growth rates of all the genera with Clostridioides across all the data sets. And what we see is that there are quite a few that actually have fairly strong Pearson correlation in their log growth rates. And so this is, suggesting that maybe there's the positive relationship here might be suggestive of some cooperation between these organisms, but the correlation only tells you so much. So to kind of dig into that further, here I'm showing you the import flux space for the entire communities across all the data sets. And here I'm also coloring these top 3 most correlated genera, as well as Clostridioides. And some of the features here we see, again, for Clostridioides, we see there are multiple clusters, again, suggesting multiple strategies. We also see that this is true for some of these genera, which are correlated with C. diff, but also we see there's very little overlap in the niche space of these other genera and Clostridioides. Again, maybe suggesting that they are not necessarily competing with each other. Maybe there's some cooperation, but not in every case. You do see there are some points where there is overlap. And so, yeah, one of the things we see, for instance, if we look at succinate import, again, one of those metabolites that is known to empirically promote C. diff's growth, what we see is that Clostridioides will generally import this metabolite when it's predicted to grow well. And when it isn't growing well, you see there's competition. In this case, specifically from Foca-Kola, which is Bacteroides vulgatus. But also you see some competition with Bladia and Raminococcus. But importantly, the relationship with Bladia is sort of context dependent, because there's both the Bladia, the Foca-Kola, Bacteroides, and a number of other genera you see when they're not competing with C. diff for succinate, in this case are also exporters of succinate in the case where C. diff's growing best. So it would seem in this case that several of these genera are potentially providing one of the key metabolites that C. diff is subsiding on. This is true for ornithine as well. For ornithine, you see the consumption by Clostridioides is very context dependent. It generally, when it does consume it, it grows very well, but it doesn't always consume it. And you see, in this case, there's a lot more competition for ornithine. You see there's a lot of genera that are also consuming it. And when they are consuming it, generally Clostridioides is not. And among those, you see again, Bladia shows up, Foca-Kola, and a few others, the Eubacterium as well. But similarly, as before, you see some of these individuals some of these genera that are competing for this metabolite are also potential exporters. And when they are exporting it, C. diff's importing it, and it seems to promote its growth. So this really just points to the context-dependent nature of these cooperative and competitive interactions and their importance in promoting or preventing colonization. And so, I'll end there with kind of some of the data that I'm showing you and discuss some of the paths forward. What I showed you are, some very interesting results that kind of point towards specific mechanisms. But, with correlation studies, these are still putative mechanisms. And so really, we would ultimately like to validate these further using very controlled studies. It's hard to do this in human populations. One of the challenges discussed today, for instance, are formulating accurate representations of the diet. And so another strategy here that we're looking to leverage are in vitro studies, where we can use well-defined and simplified communities to test some of these modeling hypotheses. We can look at communities of varying composition. We can also use well-defined media. And there are some data that have already been generated for this purpose, particularly the study that was recently published, looking at communities of different complexity and how they interact with C. diff in vitro. And importantly, like with some of the other results I showed you, we can use this modeling framework to test our predictions and compare them against the empirical growth measurements, but also potentially compare our predicted fluxes with empirical measurements of metabolite production and consumption using other omics measurements. And so to conclude, just to reiterate a little bit what I told you. Oh, it froze. Oh, there we go. Sorry. So I told you a little bit about the development of this modeling framework to simulate pathogen colonization and how I've leveraged it to look at mechanisms of C. diff colonization and how the community might be playing a role in this. But this is a fairly generalizable framework. And so some future directions that our group or maybe some of y'all might be interested in pursuing would be to leverage these methods presented here to predict engraftment of other genera of interest or species even. So for instance, here I used it to look at C. diff specifically, but you could imagine you could use this to predict engraftment of other pathogens or even probiotics. And you could also, as was presented in the course, assess the role of diet and community composition on colonization, and really potentially use this for engineering specific microbiomes for the purposes of interest. So with that, I'd like to acknowledge everyone who has supported me in this endeavor, in particular my advisors, Sean Gibbons and Nin Baliga, as well as Christian Diener, and everyone else who's worked with me. Thank you so much. So maybe now we can take some questions if there are any. Sean, do you or anybody?
Yeah, so I guess one question that came up is, things you presented, they're all using similar tools that Nick showed us today. Is that the case?
Yep. I'm using MyCom straight out of the box. And you just really the only key difference here, I'll go back to this slide, is really I just developed this assay, this kind of perturbation assay where we're introducing a non-native microbe to the community, which was represented in the data. But yeah, you can use all the tools that were presented today to do very similar types of analysis.
Yeah, one question on which shows that how to determine if something is not growing. Why you see so much higher outcome times of contribute along the platform?
Oh yeah, that's a great question. Yeah, one of the challenges we Nick briefly talked about the use of the solvers to be able to solve these very complex linear problems. And so one of the challenges there is the numerical accuracy of the solver. And so empirically, we know that the solver we've been using, one of the, I believe it's CPLEX with the academic license, it has an accuracy limit of 10 to the -6 in terms of like the numbers that it will generate. And so given that constraint, we chose that as our cutoff. So any value less than 10 to the -6 was considered 0 essentially. And so we actually, any value lower than 10 to the 6, we make 10 to the 6. And so that's why you see pile up around that value. Any other questions?
One that came up. 16S data is limited to species or genus level, but bacteria at a strain level might have different metabolites, different strains of C. diff, for example. How can Microbiome
Yeah, that's a very good question. Yeah, if we did have strain-specific data, we could investigate that. In our case, yes, we are limited by the 16S data. And so we had to average across all strains. So for example, all the genus level data that I showed today are compiled from all the strain specific models. And so even for the Clostridoides model, that's actually composed of 4 different strains, 2 of which are known to be toxigenic and 2 that are not. And so, yeah, that is an important distinction. And hopefully in the future with more accurate estimation techniques, like for instance, the use of metagenomics, you can get down to the species or strain level, but that is a limitation of our study. Sweet. So with that, thank you very much again. And I will pass it off to Sean for closing remarks.
Let's see. All right. Thanks, Alex. Really great talk. Super exciting work. That should be a preprint soon, so keep your eye out for that. So that wraps up day 2 of the course. Thanks again to our sponsors, Illumina, Metabolon, and the Washington Research Foundation. And a special thanks to our ISB team, and in particular today, Nick Bowman, for walking us through an excellent tutorial in metabolic modeling, and our presenter that you just saw, Alex Carr, for showing us another kind of use case for this type of modeling platform. To kind of recap, today you learned how to build a community scale metabolic model using microbiome compositional data and data on dietary intake. Right, modeling a single strain is quite different from modeling a complex community, and the types of tools and things you need to take into account are different. And so MyCom is specifically optimized for modeling diverse communities and looking at their functional outputs. You saw how important it was to match microbiome and dietary composition in order to get accurate estimates of functional output. And you learned how MyCom could be leveraged to simulate, the invasion of an external taxon into a microbiome, right? An organism that's not currently living in the system being brought in and asking whether or not it sticks, right? Can it colonize? Can it engraft? And this is sort of a mechanistic framework for trying to make those types of predictions, both in terms of something like C. diff, which is a opportunistic pathogen, but also you could imagine turning this approach towards probiotics and potentially beneficial organisms or fecal microbiota transplants and what have you. We look forward to hosting you here again tomorrow for our symposium. So last 2 days of coursework was to kind of give you an introduction to a few methods and techniques in the microbiome field, kind of systems biology and microbiology, with a focus on data coming from underrepresented populations. And this year we have an amazing 1-day symposium with 6 researchers from around the world who are doing research in partnership with and focusing on these populations and increasing the breadth of our understanding of the human microbiome and phenotypic diversity across the world. So we'll be hearing from those folks tomorrow. We'll kick things off at 9 a.m. Pacific time, just like we have the last couple of days. We hope to see you all there. If you want to learn more about us and ISB, you can go to isbscience.org. And with that, thank you all so much for joining us today. Thanks for your questions and for your engagement. If you have further questions, please take advantage of the Slack. And for those of you who are watching this on the YouTube channel, when you wake up tomorrow morning, please, again, use the Slack channel if you have any questions about the course exercises today. All of this material will live forever on the web, on our website. And so you can watch all of our videos and lectures and use all of our course materials well after the live events that you've seen the last couple of days. So thank you and have a great rest of your day.